Hi, I am Muhammad Shazly, and today we would like to present our project entitled Perturbed Amino Acid Metabolism Pathways in Chronically Anxious Zebrafish. So firstly, we will go to introduction part. The statistic shared by WHO stated that there are 284 million of people globally affected with anxiety disorders. The numbers comprise of 3.6% of the world population. From the numbers, there are 4.34% of the anxiety disorders cases come from Malaysians. So even though the current treatment available, the number of cases are quite large. This is because of the heterogeneity of the patient's responses on the treatment, so that urge for more studies. So in these studies, we used the radio, also known as Zebrafish, to investigate the perturbated pathway underlying anxiety on the brain of the chronically anxious zebrafish. The reason we use zebrafish in this study is because of small in size and cheap, yet the zebrafish has evolutionary conserved traits that make them suitable to make an animal model. Next, we move to the methodology part. The zebrafish firstly undergo chronic unpredictable stress paradigm. The paradigm consists of the treatment phase and the stressor phase. The treatment consists of the control group, treatment group, which is we use the fluoxetine at 5 mg per liter concentration, and lastly untreated group. The N for each group is 10. So next is the stressor. The stressor that we use a predator, warm water, tight space, and out of water. Each phase were repeated in each individual of zebrafish for 14 days. At the end of the 14 days, the zebrafish brain were obtained and prepared for the sample preparation and sample analysis for the global metabolomic using LCMS cutoff. So the result that obtained from the sample analysis from the LCMS cutoff were further analyzed of its pathway analysis. So next we move further. Here is the result of this study. The table shows the selected metabolites and the impacted pathway involved. So the selected metabolite were based on the score of area under curve, which more than 0.75. So the metabolites that were selected are inosine 5 monophosphate, spermine, spermidine, and L-glutamate. The impacted pathway that involve are purine, glutathione, arginine, proline, as well as D-glutamine and D-glutamate metabolism. As we can see, zebrafish that were treated with 5 mg per liter concentration of the fluoxetine has upregulation of all selected metabolites. The high concentration of the metabolite important in the regulation of the pathway involved, which important from protecting from the oxidative stress. So the oxidative stress has been reported has association with the anxiety disorders and other mental disorders. So in conclusion, we had successfully identified the perturbed metabolism in the anxious zebrafish, chronically anxious zebrafish, and the pathway that involved. So zebrafish can make a suitable model in future for the metabolomic study that relate with the anxiety. So before the end, we want to thank the Best Study Grant, UiTM, for the funding, and also the iPROMISE for providing the Zebrafish facility. So these are the references that I used in this study. That's all from me. Thank you.